Paul?
Okay, should we?
Yeah, sure.
All right, let's get started. Yeah, thanks again, Dr. Stu Tangye, to accept our invitation to give a wonderful talk. Before your talk, I would like to introduce you to our audience so that people will know you much better. So it's really my honor to introduce Dr. Stuart Tangye, a well-renowned immunologist and then a leading figure in the field of human immunology and inborn errors of immunity. Dr. Tanya has earned international reputation for his groundbreaking research, which focuses on unraveling the molecular and cellular mechanisms behind immune dysregulation. His work has profoundly advanced our understanding of how genetic mutations affect the lymphocyte differentiation and function, contributing to conditions such as primary immune deficiencies, autoimmunity, allergies, and cancer. Through his innovative studies, Dr. Tanya has identified critical pathways in immune responses, opening new avenues for therapies to treat immune-related disorders. Dr. Tanya's distinguished career includes his current role at the Garvan Institute in Sydney, Australia, where he leads a highly productive research lab. His transformative work has been instrumental in identifying and characterizing novel inborn errors of immunity caused by mutations in genes such as SH2D1A, STAT3, and DOCK8, among many others. In 2015, he founded a clinical immunogenomics research consortia of Australasia, a pioneer initiative that uses advanced multi-omics technologies to diagnose and improve care for patients with rare immune diseases. Since March 2020, Dr. Tanya has served as a leader of the Oceania hub of the COVID Human Genetic Effort, a global consortium that has made groundbreaking discoveries about the role of type 1 interference in defending against SARS-CoV-2. His leadership and his effort has been critical in enhancing our understanding of the immune response to viral infections. With over 260 peer-reviewed publications and more than 35,000 citations, Dr. Tanya's contributions have had a profound impact on human immunology. And today, I'm thrilled to hear more about new discoveries from Dr. Tanya, and let's give him a warm welcome. Please, Dr. Tanya.
Thanks very much. Thanks for that very, very generous introduction. It's lovely to be here. And congratulations to the team behind the Immune Zoom seminar series. I think it was a great initiative that you set up when COVID hit us all. It's a great way to keep the community connected and get to hear some science when we were locked down or unable to travel or even unable to come to work. So as Paul said, I'm going to talk today about a lot of the work my lab's been doing. I'll summarize a large chunk of the work briefly in the first part, and then go into some unpublished work that's sort of happening as we speak in the lab currently. So I have no conflicts of interest or declarations to make, except that I'm thrilled to be here, and it's exciting to be part of this seminar series. So I'm going to introduce a couple of concepts or ideas or fields first, and then bring them together to tell you how that sort of guides my research. So as Paul mentioned in my introduction, I'm really interested in this set of diseases called inborn errors of immunity. And these are diseases that are what's called monogenic. So they result from a defect in a single gene. And as I've got listed there, it's a germline gene. So people are born with these mutations. They're not somatic, they're germline. And they affect the function of a single protein. And so, we've got 25,000 genes in our genome, and a mutation in just a single one is enough to be problematic. And that's because the variant in the gene results in the protein either becoming a loss of function or a gain of function. The protein can function as a dominant negative protein. It can be haploinsufficient, or something we're seeing a little bit more now of, and I suspect we'll see a lot more of this in the future, is that the protein actually becomes neomorphic. In other words, it acquires functions that the wild type protein does not have. And that structural or biochemical or functional change to the protein is enough to cause a human disease. We know there's a lot of redundancy in the human system, but these highlight the fundamental requirements of these specific genes for, in this setting, immune function, immune homeostasis. Typically, we think of inborn errors of immunity as manifesting as severe infection, and that's certainly true. Many inborn errors present initially with recurrent or severe, sometimes life-threatening infections, but the phenotype of these conditions is much, much broader. It includes autoinflammatory, autoimmune conditions, allergic disease, atopic disease, and even cancer. And sometimes the one gene can cause all of these phenotypes, not necessarily at the same time, but throughout the natural history of a disease. Uh, Patients can present with all those different features. As I said, these are germline variants, so consequently, they can present early and, in fact, very early in life, even at birth. And these can be fatal, tragically, but by and large, they definitely have a significant impact on the quality of life. And this is just to give you some examples of what these diseases can look like and can include. On the left here, this is David Vedder. You probably know him better as the boy in the bubble. He lived about 12 of his 12 and a half years in a sterile bubble. And that's because his immune system was was unable to protect him against almost any pathogen. So bacteria, viruses, parasites, fungi, all of those infections he was susceptible to. And that's because he had a mutation in the IL-2RG gene. That's the common gamma chain component of the IL-2 family of receptors. So he was susceptible to everything. But then this person here, who has a mutation in one of the receptors for IL-17 family cytokines, they only have susceptibility to fungal infections, like not great, but you can see this pathway is really only needed for protection against antifungal immunity. As mentioned, these can be inflammatory diseases, and this is probably one of the best examples of a monogenic severe inflammatory disorder due to a lack of IL-10 or IL-10 receptor signaling, resulting in inflammatory bowel disease. You can have infections that are very much anatomically restricted, so the DOC8 deficiency really results in very horrible skin viral infections. Similarly, CD28, which I guess strikingly and surprisingly rendered admittedly only a few people reported so far who have mutations in CD28 that's susceptible to human papillomavirus, causing warts, and obviously very, very severe warts, but perhaps not as broadly infectious susceptibility as you may think. And this last picture indicates that someone who's got a mutation in STAT6, and this is a gain of function. So constitutively or accelerated activation of the STAT6 protein, resulting in eczema. Probably not surprising knowing what STAT6 does in terms of IL-4 and atopic diseases. Now, these are a whole collection of diseases with very different phenotypes in some cases, and there's many of them. So over the years, we've seen this huge explosion and increase in the absolute number of inborn errors of immunity. And currently, there's more than 560 genetic causes of different diseases that are known. So individually, these diseases are very rare. So each of these cases here that I show you are fairly rare, but when you add them all up, they can be much more frequent than you think. So this is an example of a fairly rare disease. This was results from the US after they rolled out the newborn screening program to look for patients who were T-cell deficient at birth. So this is screening for a severe combined immunodeficiency, which is ostensibly defined as having no T cells. So this was done on healthy babies, the first 3 million babies born. And what they found was that on average, about 1 in 60,000 births resulted in T cell deficiency or very, very few T cells. This differed across the states, so across the regions in the US. There's a well-known founder effect in the Navajo Nation First American population, where the incidence is about 1 in 3,500. But then in other parts of the US, Texas, Wisconsin, and so on, the frequency is much lower, but on average, it's about 1 in 60,000. That only measures T cell deficiency, though. A more recent study, which was published last year, which is more broadly looking across the US population using electronic health records to record symptoms and features of inborn errors of immunity, found that the incidence was more like 1 in 2,000. So again, still a rare disease being defined as having an incidence of less than 1 in 2,000, but clearly much more common than we may think when you think of an individual gene. Putting them all together, it's quite frequent. Okay, so I think based, and of course I'm biased to say this because this is what I work on, but I think it's safe to say that studying inborn errors of immunity has really led to some major discoveries and paradigm shifts in our understanding of basic clinical and translational medicine. And because the immune system is really pervasive across almost every medical discipline, whether it be pulmonology or neurology or hematology, rheumatology, and so on. It's really important to appreciate that these patients can present in any of those clinics, and by studying immunology, immunogenomics, can really inform diseases that people may not appreciate are actually immune mediated, but that's because they present as inflammatory bowel disease or meningitis and things like this. Okay, so the second topic to introduce, which really doesn't need introduction to this audience, is my other passion, which is B cells. So, as you know, B cells are central to the mammalian immune response. They do lots of cool stuff, but one of the most important functions, of course, is the production of antibody. And that antibody production is important for protection against infectious diseases and also the success of most vaccines. So the way B cells function, again, you know this, they come out of the bone marrow, enter the periphery and circulate. And then when they encounter their specific antigen, they undergo a number of activation steps. The first wave would result in production of short-lived plasmablasts, which offer some protection in the form of secreted, generally low affinity IgM molecules. But then activated B cells, particularly under the influence of T cell help, can seed a germinal center, undergo many rounds of cell proliferation. This is the classic McFarlane-Burnett's clonal selection theory in action. And out of the germinal center, again, following interactions with T cells, you can generate plasmablasts, which go on to become long-lived plasma cells. They can move to different tissues. Or you can generate memory B cells, which are also long-lived, but these cells can re-enter a germinal center or be reactivated when you re-encounter that initiating antigen. And studies done 15 to 20 years ago really showed that the half-life of serum antibodies can range from 10 to 10,000 years, while memory B cells to a specific antigen can persist for decades, up to 50 years of a host. Now, the importance of generating these types of cells is abundantly clear, and in 2025, we know that's really important. But we've known this for actually centuries, if not millennia. And I just want to show you one example of this. Oh, sorry, I'll come to that in a second. The way this works is, of course, you get induction and alteration, regulation of expression of a number of key transcription factors, which are important for inducing class switching, inducing germinal centers, inducing differentiation to these different types of B cells. So we know it's really important to generate these B cell types, and we've known that for centuries, if not millennia. But this is just a really good example of how effective and how important it is to have immunity against existing or emerging infectious pathogens. So going back a couple hundred years to the late 70s, the Faroe Islands, which are in the North Atlantic here, positioned between Scandinavia and Iceland. Faroe Islands are actually a colony of Denmark. Back in the late 1700s, when there was a population of under 50,000 people, there'd never been a measles. But then in 1781, there was a measles outbreak in the Faroe Islands. But then measles wasn't seen again for 65 years. Tragically, or unfortunately, for a visitor in 1846, they arrived on the Faroe Islands and actually brought measles with them. And what was remarkable was that anyone who was over 65 who was infected back in 1781, they didn't become sick. They got infected, but they actually didn't become dreadfully unwell. And you can find papers which talk about here that none of the 98 aged people who were around at this time and who had measles were significantly affected by infection. They were infected, as in measles positive, but were not symptomatic in any major way. So again, just shows the longevity of immunity, even in the absence of exposure to the same antigen for 65 years in this case. The first exposure still left that significant imprint on the immune system for immune memory. Okay, so when I was doing my PhD, which was back in the early 90s, so 30 years ago now, 30 plus years ago now, I was really inspired by a lot of work that was going on at a couple of research institutes. They're both listed on many of these papers, actually. One was in France, in Lyon, Dardilly in France, and the other was in California. at the DNAX Research Institute. And at the time, labs in these places were studying the effects of cytokines on B cell behavior. So I was really drawn to this work. I was really interested in this work, and I ended up going to DNAX to do my postdoc in the mid to late 90s. But I was really curious about what cytokines can do to B cells and how B cells can become activated. And it's really been a theme of my work ever since. So we know cytokines are important for B cell function. We also know T cells are important for B cell function. And again, that's obviously not a new finding. We've known since 1965, at least. This is lovely work done by Jacques Miller, showing that if you take a thymus out of mice and then immunize them, in this case it was with salmonella, if you don't have a thymus, you don't generate what is considered an antibody response compared to the intact mice. So we know that T cells are important for generating antibodies. We know that cytokines can influence antibodies. So that's something that I've been really interested in. And again, these studies obviously came together when people did these sorts of studies, these sorts of publications, again, back in the 80s and 90s, showing that these interactions between CD4 T cells and B cells, either both at the cell surface level and in the secreted form with cytokines, are important for activating B cells. So this is sort of what I've been studying for a long, long time now, trying to dissect what are the relative inputs and hierarchy of signals that are needed by B cells for them to become useful and helpful and effector cells in the settings of immunity against different sorts of pathogens and vaccines. So more than 20 years ago, my lab started looking at the effects of IL-21, interleukin-21. Today, it's by no means a new cytokine, but it was back in the early 2000s when it was first discovered. It was first described as an important factor for T cells and NK cells, actually. But then my own lab and several other labs around the world really did a lot of interesting and important work dissecting out what IL-21 can do to human B cells. And this really summarizes a lot of publications. Some are shown down here, showing that IL-21 is actually a fantastic growth and differentiation factor for human B cells. So in vitro, when you activate B cells with T cell help in this form of CD40 ligand, in the presence of IL-21, these B cells undergo massive proliferation. So that's a bit like that clonal expansion I was talking about in vivo. They can undergo the key events of humoral memory. So class switching, B cells can class switch to IgG and IgA, but then they can also become antibody secreting cells, plasma-like cells in vitro, this is, producing all of the immunoglobulin isotypes. And these processes can be remoted or reduced in the presence of other cytokines. So IL-4 promotes IgG, IL-4 promotes IgE with IL-21, and things like this. So there's this real dominance of IL-21 being able to induce all of these processes. And the reason this happens is because, like the germinal center reaction, IL-21 is capable of inducing or modulating key molecular mediators of those processes that are shown here. So IL-21 induces BCL6, which is the signature transcription factor of germinal centers and germinal center B cells. Also induces AICDA, which is AID, required for class switching, and also induces regulators of the plasma cell pathway, BLIMP1, XB1, and downregulates PAX5. So in one cytokine, you get this whole molecular program induced, which in different groups of B cells, depending on stochasticity or signal strength, can become class switch B cells or antibody secreting plasma cells. So that led to a whole lot of work, which I'll get to in a second, but it gave us the idea that how can we look further into mechanisms of IL-21 function in humans, and what tools and resources do we have at our hand? So just jumping back to my postdoc days, I did my postdoc with Jan de Vries at DNAX in the, as I said, mid to late 90s. And one of the papers that I was lucky enough to publish there was this one here. And what we found is that the molecule CD27 is actually differentially expressed on human B cell subsets. So it's absent from naive B cells. It's expressed on plasmablasts and plasma cells, and it's induced at the germinal center stage and remains on germinal center B cells as they become memory B cells. And you can see this is real data, as opposed to my cartoon here, showing RNA-seq data and FACS data, flow cytometry data, this nice graded increase in CD27 expression as the naive B cells progress through these differentiation stages. And in this paper, I just wrote in the abstract or somewhere along the way that assessing expression of CD27 may be a convenient way to monitor the development of memory B cell responses in immunocompromised individuals. That was in 1998. And I'm pleased to say, but also surprised to say, that's a thing we've been doing for decades now. So um, we know that IL-21 is a cytokine. It signals through its cytokine receptor, and as everyone here would know, cytokines generally signal through JAK-STAT pathways, and JAKs are kinases which associate with cytokine receptors. They become activated and phosphorylated when receptors are ligated, and then they can activate and recruit STAT molecules, which become phosphorylated dimerize and translocate to the nucleus, where they regulate gene expression. And over the last 25 years or so, inborn errors have been reported to be caused by mutations in almost all of the JAK-STAT proteins and genes that we have been identified. Please don't labor too much on these orangish looking boxes with the text. It just highlights that there's a whole series of patients out there who have single gene mutations in JAKs and STATs, which result in different types of diseases. But this actually gave us a huge opportunity to understand how does IL-21 regulate B cells in vivo if we could study B cell subsets and B cell function in patients with different inborn errors of immunity affecting cytokine signaling. So that's what we just started doing. So we know that IL-21 binds its receptor complex, which is comprised of a unique IL-21 receptor chain coupled to the common gamma chain, activates JAK1, JAK3, STAT1, STAT3, and STAT5. So we started looking at patients for memory B cells in their peripheral blood using CD27 as a marker of memory B cell formation. So if you look in the blood of healthy donors, about 25% of our B cells have CD27 expression. And when we looked in the first set of patients, these are patients who had mutations in STAT1 or mutations in STAT5, you can see here that really the memory B cells are not significantly affected, not reduced. In fact, they might be a little bit increased in some of the STAT5 deficient patients. But really, STAT1 and STAT5 had no significant or not an impressive effect on memory B cells in these patients. However, when we looked at patients with STAT3 mutations, we could certainly see a significant and consistent reduction in memory cell phenotype cells in the blood of these patients. And we've seen this in probably over 50 patients by now. Now, that's interesting, but as you can appreciate, STAT3 is downstream of a lot of cytokine receptors. It doesn't actually tell you which upstream cytokine receptor is the one important for this process. But as time moved on, more and more patients with different mutations were identified, and we were able to look at some of these individuals. So the first group we looked at were patients with mutations in the IL-2RG gene. Now, this does cause X-linked severe combined immunodeficiency, and those patients usually go on to get transplanted. But there's a group of patients who, if they're not conditioned pre-transplant, they actually retain their own B cells. So these are boys who have their undergo transplant but retain their own B cells, which contain the mutant common gamma chain gene, but they're reconstituted with T cells from a donor. So they're like a mouse bone marrow chimera where their B cells remain to be defective in IL-2RG, but almost every other immune cell type is reconstituted. So even in the face of T cells that have wild type IL-2RG expression, the memory B cells remain reduced in these patients. And then we could confirm that was very likely to be due to IL-21 receptor when Gilbert-Uzel and others identified IL-21 receptor deficient patients in 2013. We could show that there's a memory B cell deficit in those patients as well. Now, interestingly, we've looked at a whole suite of patients with mutations in other cytokines or cytokine receptors or JAK-STAT molecules, and there's possibly a very modest role for IL-6 and IL-10, but there's no role for those other cytokines that I've mentioned there. Now, interestingly, we've focused on B cells here, obviously, but it's well worth pointing out that some patients with some of these mutations also have defects in the right type of helper T cell that's important for humoral immunity, and they are the T follicular helper cells. You can identify these cells by expression of CXCR5 amongst the peripheral blood circulating T cells. And you can see patients with mutations in STAT3 or IL-21 receptor have fewer of these cells. And importantly, these T cells have reduced capacity to produce IL-21 when cultured in vitro. So there's clearly a B cell defect, but there's probably a T cell defect in being able to provide help to B cells as well. So just extending this a little bit further, we wanted to look at class switching, and that's because, as I said, IL-21 is capable of inducing AICDA, the major requirement for class switching and somatic hypermutation, but in this context, class switching. So what we next did was just look at the memory B cells in many different types of patients with these known gene defects and look for what proportion of the memory B cells had undergone class switching. And you can see that similar to layout to what I showed you before. Interestingly, you can actually get class switching even if you have a STAT3 mutation, but it's the patients with mutations in IL-2RG, IL-21 receptor, who have very few class switched memory B cells. And the level of these cells is approaching that what we see in patients who have AICDA mutations, AID deficiency, who cannot undergo class switching at all. So that tells us that IL-21 signaling through its receptor is really important for inducing class switching. Now, that's all ex vivo flow cytometry that I'm telling you. So it is possible that this is all secondary to a defect in T cell help. But we really didn't think that was the case. So we look at B cell function in vitro. We're looking at antibody secretion here. So this is IgM and IgG, production by B cells from healthy donors, patients with these different mutations, STAT1, STAT3, IL-10 receptor, and other components of the IL-21 receptor pathway. And you can see that in response to IL-21 stimulation, there's very little induction of IgM or IgG by patients with mutations in STAT3, IL-2RG, IL-21 receptor, as well as another transcription factor that regulates STAT3 function. Likewise, you don't get induction of BLIMP1 in patients' B cells with mutation in STAT3 or IL-21 receptor. Again, showing that that whole molecular machinery that's required for driving B cell differentiation in vivo and in vitro is affected by STAT3 mutations downstream of the IL-21 receptor. So just to summarize that part of my talk, what I've shown you here is that whilst B cells, and I showed you that slide early on, a lot of in vitro studies, work done in the 80s and 90s showed that many cytokines can actually activate human B cells in vitro, and they can induce changes in B cells regarding phenotype, proliferation, antibody production. But so IL-21 is the fundamental requirement to kick that process off. And you need IL-21 provided by CD4 T cells, these T helper-like cells, signals through IL-21 receptor. And without that, none of these other cytokines are able to compensate for that dominant role of IL-21. And what then happens is you get the production of plasma cells and class-switch memory cells, which are really important for that long-lived protective immunity against infectious diseases and in response to vaccination. So the second part of my talk is sort of an extension of this. I'm going to talk a lot more about STAT3 deficiency now and the disease that it actually causes, which is called autosomal dominant hyper IgE syndrome. So this syndrome is actually first described in the 1960s, but it wasn't until 2007 that the genetics underlying hyper IgE syndrome were revealed, and that was done by Steve Holland, Bodo Grumbacher at NIH and in Freiburg, but also by Yoshi Minagishi in Japan, who identified dominant negative heterozygous mutations in STAT3 cause this syndrome. Now, this is a multisystemic disorder. There is obviously an immune component. They have poor antibody responses. They have susceptibility to some pathogens, fungal infections, bacterial infections like staph and strep. As the name suggests, they have hyper IgE, some eosinophilia, but they also have problems with non-immune tissues. So they have musculoskeletal defects, connective tissue defects. They retain their primary teeth. They also have vascular defects. And there's also some issues to do with viruses and B-cell lymphoma. So this is what STAT3 looks like here. You can see the mutations are predominantly located in the DNA binding domain, the SH2 domain, but also linker and these transactivation domains. Very few mutations affect the N-terminus, which is interesting in itself. But this is the types of mutations that have been reported. They're mostly missense mutations where you have introduction of a different amino acid, or sometimes there's a triplet deletion where you lose a particular amino acid. Over the years, much of the mechanisms underpinning some of these clinical features have been identified. So work that I just showed you then, and other work done particularly by Cindy Ma's lab, who is one of my collaborators here in Sydney. We've shown the molecular cause for the B-cell defect is probably IL-21. IL-10 may have a contributory role there. Some work Cindy did a long time ago showed that the infectious susceptibility is due to a Th17 deficiency, so a defect in generating effective T helper 17 cells. Other work done by others have shown that the musculoskeletal defects are probably due to IL-6 and IL-11. And there's also some defects in CD8 T cells. But what I'm really fascinated is by this, what I think is an immune paradox in STAT3 deficiency. So as I've told you, and I'm going to show you again here, these patients have well-characterized, well-reported defects in generating antibody responses to specific pathogens or specific vaccines. And this is a study that goes back more than 30 years, and this is well before we knew what caused autosomal dominant hyper IgE syndrome as being STAT3. But this is a lovely study showing that when you take patients and you vaccinate them with this bacteriophage called Phi-X174, you can see that in healthy donors, shown by the stripy lines here, that's what your primary antibody response looks like. And then when you come back a month later and boost those healthy donors, the level of that antibody goes up, as you'd expect, as a recall or a secondary or an anamnestic recall memory response. When you look at the patients, though, here's the primary response. For some individuals, it sort of gets within the normal range, but most of it sits below the healthy range. And then when you boost these patients, it pretty much looks like a primary immune response again. You don't have that lovely amplification in antibody titers that you would see in the healthy donors. So they're clearly immune deficient. And the data I just showed you reminds you here that they have very few memory B cells. B cells don't respond to cytokines, IL-21 in this case. And this is some data we published 15 years ago now, sort of updating studies here, but this is using patients known to have STAT3 mutations, measuring antibody levels against tetanus toxoid. And as you can see here, the titers drop off much, much more quickly than you see in healthy donors. So there's 50 to 100-fold difference in the level of protective antibodies in these patients. But on the other hand, they have, it's called hyper IgE syndrome. And that's because these patients have truckloads of IgE in their serum. And that was first reported in 1972. Interestingly, it wasn't reported in the first cases of these patients, which is actually called Job syndrome. That's because IgE itself wasn't discovered until a little while after this first paper. So in 1972, Becky Buckley identified these patients as having hyper IgE. And again, this is data from Cindy Ma's studies showing indeed STAT3 deficient patients have very, very high levels of total serum immunoglobulin. And it's probably not benign IgE because we can detect reactivities against specific allergens, in this case, food allergens. So they make loads of IgE. So this is the immune paradox that I'm talking about. If you don't have fully functional STAT3, you don't form memory B cells properly, and you have low levels of antibodies against low levels of protective antibodies. However, if you don't have fully functioning STAT3, you get tons of IgE. So the phenomena of driving class switching or driving memory cell formation or plasma cell formation to all of the isotypes is actually different. What drives IgM, IgG, and IgA is shared, and that's dependent on STAT3. But what drives IgE is actually regulated by it's regulated by STAT3. So we've been interested in this for a long time now and trying to understand what are the drivers of the IgE dysregulation in STAT3 hyper IgE syndrome. This is very difficult to do because it's very hard to find IgE positive B cells in the peripheral blood of healthy donors. And this is a multi-parameter high-dimensional flow cytometry plot showing expression of IgE on B cells from healthy donors versus IgG. So obviously we can detect tons of IgG positive cells, but not a single event suggesting IgE positive cells in the blood of healthy donors. And that's not really surprising because there was this really nice paper published a couple of years ago, which tried to do something similar. And this paper really highlights the pitfalls that can happen if you're just relying on one approach to identify these cells. So this paper basically isolated B cells based on expression of IgE, but then when they did the molecular amplification of the IgE or the epsilon transcript, they found none in, generally speaking, healthy donors. But a terrific PhD student in our labs, Kanan Pathmanandavel, developed a very sensitive flow cytometry assay, which when applied to patients with STAT3 mutations could actually detect a couple of these positive cells well above the levels that we saw in healthy donors. And we could validate that that was intrinsic expression that wasn't due to capture of serum IgE onto FC receptors or anything like that. So this actually gave us an opportunity now to really look into the nature of these memory B cells in STAT3 deficiency by a number of novel approaches. The first one being, of course, single-cell RNA-seq. So Kana and Reynolds assays looking for transcriptional signature at the single-cell level of Ig epsilon expressing cells. So these are UMAP plots showing this is the population of B cells that are expressing the epsilon chain of immunoglobulin. They also express CD27. They express high levels of alpha receptor and high levels of CD23, or the low affinity receptor for IgE. Looking a bit further at that population, we can identify a bunch of markers which are highly expressed or dramatically reduced in these IgE expressing B cells. So this confirms the FC epsilon R2, which is CD23 expression, very highly expressed in B cells expressing Ig epsilon, much higher than B cells expressing any other isotype. Conversely, though, there's downregulation of a bunch of genes which are involved in inflammatory responses, trafficking, and survival, such as the S100 proteins, CXCR3, and the RGS2, which regulates G protein coupled signaling. So we're sort of coming up with this transcriptional signature of what IgE expressing B cells looks like. Still a long way to go, but it's making progress, which is lovely. And also what's interesting is by looking for the specific immunoglobulin heavy chain genes, we could say quite confidently that the B cells expressing the epsilon transcripts have a very similar gene profile to that's seen in IgG1 positive B cells, or the gamma 1 transcript positive cells. Certainly not saying that one gives rise to the other, G1 becomes IgE, but that's very possibly the case. As we know, B cells can sequentially class switch. But it just shows you that there's probably some relationship between these cells. So basically, we now have this really good opportunity to drill down and look at these cells in much more detail, which we've started doing a little bit of, and I'll show some of that data next. That top half of this slide, I know, is very, very busy, but what I'm trying to show you here is that the first thing we've got a really solid answer for is somatic hypermutation. And this is looking at somatic hypermutation in all of the heavy chain transcripts of B cells from healthy donors. And I know it's very hard to see, but this is the few events you get for IgE. There is some level of somatic hypermutation. And this is what we saw for patients with STAT3 hyper IgE syndrome. It's probably much easier to look at this summary plot here, where I've just condensed this down to the averages. So you don't get much mutation in B cells expressing IgM or IgD because they're mostly naive B cells. But you can see the level of somatic hypermutation in the IgE B cells is similar between patients and healthy donors. And it's also within the range that you see for other immunoglobulin isotypes. So it shows that whilst there's abundant production of IgE, it's actually somatically mutated, suggesting that some degree of selection or affinity maturation. So looking a little bit more in detail here, we want to try to have an understanding of what's dysregulating IgE. So there's a set of other immunogenic disorders which affect the IL-6 pathway, which is upstream of STAT3, which also have hyper IgE phenotype. So patients who have mutations in GP130 or in the IL-6 receptor alpha chain, they come together to form the IL-6 receptor, or mutations in STAT3, as I've already shown you, or in this transcriptional regulator of STAT3, ZNF341. Those patients have all been identified, and those patients all have hyper IgE syndrome. So I've shown you the data for STAT3 already, but these are these other genotypes here, ZNF341 and IL-6ST deficiency, showing these very high levels of IgE, as well as the allergen specificity of these IgE molecules. So what that suggests to us, at least, is that likely IL-4 and IL-13 drives this hyper IgE production, but somehow IL-6 receptor signaling is what acts as a handbrake for IgE production. So I've looked at this in a bit more detail as well, coming back to this somatic hypermutation analysis, single-cell RNA-seq data from patients with all these different genotypes, as well as patients who just have regular atopy. You can see the level of somatic hypermutation in the IgE epsilon chains is pretty comparable to healthy donors across the board here. So what we think we're seeing here is the generation of IgE positive B cells in hyper IgE syndrome, which are probably similar to IgE positive B cells in healthy donors or in common diseases like atopic diseases. And that gives us a model or an opportunity to really understand the transcriptional programming, the molecular regulation of dysregulated IgE production in these monogenic forms of atopic diseases. So basically, what we think is happening here is that you need STAT3 to give you protective immunity. That's pretty clear. But you also need STAT3 to rein in dysregulated production of IgE and to temper some of those responses that may be happening in the setting of atopic diseases, Th2-driven types of pathologies. But that also raises the question of a lot of the data I showed you around this part of the talk is all derived from polyclonal study analyses of memory B cells and antibody responses in vitro in response to mimics of T cell help. But what happens in the setting of a real immune response? And for better or for worse, the SARS-CoV-2 pandemic provided us an opportunity to look at this in a bit more detail. So for the last 4 years or so, we've been tracking immune responses in patients with different types of immunodeficiencies following natural infection, or more usefully, at least from our Australia's perspective, given we didn't have a lot of SARS-CoV-2 in the first wave of the pandemic, following vaccination. And this has been a global effort with collaborations with colleagues in the US, in Europe, in Japan, and then many, many clinicians across Australia. So we're basically collecting blood from patients whenever we could over the last 3 to 4 years in this in post-vaccination or post-infection. So firstly, I just want to set up the system. This is how we study this. We measure spike binding B cells. So this is using probes, which are derived from spike and the RBD peptide of the spike protein. Use these to detect ex vivo spike binding B cells. And you can see here in the green line, these are individuals who were SARS-CoV-2 naive at the time of assay. So their spike-specific B cells were very low, increased over time following vaccination, and would go up again every now and again with additional vaccine doses. People who were infected before vaccination, not surprisingly, as you'd expect, had a higher baseline level before vaccination, and these B cells went up and came down, and eventually they sort of reached the same level after multiple vaccine doses and, of course, time. These two graphs down the bottom show antibody levels to total spike, and this is neutralizing antibodies to spike proteins. And again, follows similar patterns to the B cells, as you'd expect, based on classic immunology textbooks showing the kinetics of antibody responses. So that's in healthy donors. What happens when we look at patients? So, firstly, I'm just going to show you a little bit more data, and this is data from another PhD student in the lab, Joe Mackey, who has done quite a lot of this work for his PhD, but in other aspects of the projects as well. This is just showing the phenotype and the nature of the SARS-CoV-2 specific B cells in healthy donors. So what Joe has shown here is that pretty much early on, within weeks and months of the first and second vaccine dose, most of the SARS-CoV-2 specific B cells express CD27, that memory B cell marker. Level of class switching is evident very early on, but tends to increase. You can see the purple bar here. Early on, about half of the spike-specific B cells are IgG positive. And then after a couple of doses, this is more like 75%. And what's really interesting is early on, there is a proportion of responding B cells that have this phenotype that's of CD21 low. You've probably heard of atypical memory B cells. And I'll come back to these in a little while. So this shows the responses in patients with STAT3 deficiency, tracking spike binding B cells during the first 3 doses of vaccine. And overall, you can certainly detect spike-specific B cells in most of the patients tested, which is quite interesting because there's clearly a defect in humoral memory. But what was interesting is that the phenotype of these B cells was quite different to what we saw in healthy donors. So this is like what I showed you before. When you look at spike-binding B cells in healthy donors, most of them express CD27 and IgG, and that's shown in these two graphs here. 60 to 80% of the B cells are CD27 positive and IgG positive. However, when you do this for patients with a STAT3 deficiency, which is shown here, very few of the B cells become CD27 positive, and a good proportion, but not as many, switch to IgG. So we wanted to look at that in much more detail. So Karnan, again, developed this globalized clustering. This isn't single cell RNA-seq data. It's single cell, but it's flow data, looking at IgG negative B cells. And when you break this down based on phenotype, you can pretty much detect 6 clusters of cells, as shown here. You can see these top 2 clusters are sort of classic memory B cells. These two in the middle seem to be related, but they seem to be early forms of memory B cells. And these last two correspond to this CD21 low population of B cells. And what we can do with that, of course, is then lay over where the SARS-CoV-2 specific B cells sit from healthy donors and the patients. So this is what I showed you a second ago, but the black dots now are where the SARS-CoV-2 specific B cells sit. So for healthy donors, not surprisingly and reassuringly, they have a phenotype of what we call classic memory B cells. They sit where IgG memory B cells would. Some have acquired CXCR3 for trafficking. And there's this other cluster here, this dark blue cluster here, which have features of memory cells such as IgG. They just haven't switched on CD27 yet. However, when we looked at STAT3 deficiency, what we saw was quite different. The distribution of these black dots was quite distinct from that from the healthy donors. We see a lot of cells sitting in this red zone here, in this light green zone here, and also some cells in this blue space. So they have this in common, but these are the dominant cells for the STAT3 deficient patients. And to hopefully put that in slightly better context, you can see that if you look at the clusters 1 to 6, healthy donors sort of sit in the first 3. The STAT3 deficient patients' B cells look like they do in the last 3 populations there. So just to expand that a little bit and head towards the end of my talk, what we think is going on here is the STAT3 deficient B cells are acquiring this phenotype of CD21 low B cells. And this is an interesting population of B cells, which we've known about for a long time, but they're a very enigmatic population of cells. So we know how naive B cells behave and respond and give rise to memory B cells and plasma cells. But there's this other population, these CD21 low B cells. They have many different names. They have different transcription factors involved in this process. We don't really know where these cells come from exactly. We know a bit about them. They comprise about 2 to 3% of your peripheral blood. They are dependent on the transcription factor TBET in humans and in mice. This is. We know that they can come from naive B cells in vitro, and STAT1 and some other STAT1 activating cytokines are required for this. But we really, and there's other data showing that they can give rise to pre-plasmablasts quite promptly. However, it's also quite clear that these cells actually don't make much antibody. And this is data from our own lab, done by Danielle Avery in my lab, showing that if you sort transitional naive CD21 low B cells from healthy donors or memory B cells, they don't make a lot of antibody in vitro. And this is data in response to OFF21. See the same response, though, if you look at other types of stimuli. So there's a lot of questions that are unknown, but this brings us back to the question of, okay, we can see some B cells that are spike specific in the STAT3 dominant negative patients. Their phenotype is atypical. What's happening to the antibody response? And this is what we see for one STAT3 patient who we've tracked for a very long time now. And this is neutralizing levels of antibody to spike protein. And you can see the response is very delayed. It does come up, but it wanes. It goes up again with boosting, but then it just sort of sits and still remains. It's quite significant, obviously, but it sits quite below what we see in healthy donors. And this is a log scale, so this is quite a reduction. That's one patient, just because we've tracked that patient for a long time. But you can also see it again when we put the data together for all of the STAT3 deficient patients we've looked at, pretty much the same pattern. It's delayed, it does come up, it drops off. Many patients remain negative or undetectable levels. It moves up again with boosting. And eventually with repeated boosting, you do sort of get to those levels that you want to see, but it takes time and it takes repeated boosting. But the unknown, of course, is what those CD21 low B cells are actually doing in that space. So just to conclude that part of my talk and my talk entirely, I work on STAT3 dominant negative hyper IgE syndrome has really shown that you need this pathway to generate memory B cells. We've been able to identify this really interesting population of IgE expressing cells, and that's going to help us really understand how that pathway is regulated, and in this case, dysregulated, leading to aberrant production of IgE and allergen-specific IgE. By looking at response to vaccination, yes, you can generate spike-binding B cells in STAT3 deficiency, but they had a phenotype which is quite distinct from healthy donors. They look like these CD21 low B cells, which we know are not very helpful. And over time, you can induce neutralizing antibodies, but it's delayed. Titers are lower, and it wanes more rapidly but can be maintained or improved with boosting. So I just want to stop there. Thank everyone who's been part of this work. These are the people in my lab. So I mentioned some of the people involved in this work, but it's really been a team effort by many people. A lot of my work's done in collaboration with Professor Cindy Ma, who's my lab neighbour. And so Cindy's lab members in her team have contributed to a lot of this work. I work a lot with Chris Goodnow's team here, colleagues at different institutes in Sydney, particularly Stuart Tangye and Fabian Brolo, who have done a lot of the SARS-CoV-2 antibody work with us. Many of the clinicians nationally and internationally, without whom I couldn't have accessed any patients that have done any work, and our funding, Australian government, the Job Research Foundation, and the Allergy and Immunodeficiency Foundation of Australia. And I'll stop there and happy to take questions if there's time. Thanks very much.